All right, hey guys, welcome to our Unit 3. Unit 3 is called Cellular Energetics, and we're going to be learning about enzymes, environmental impacts on enzymes, cellular energetics, photosynthesis, and cellular respiration. So there are 5 topics in this unit, and we're going to kickstart with enzymes. All right, here we are, our first topic in the unit, enzymes. So we're going to start off with what a metabolic process is. And I've used this word a few times, and I'm pretty sure you guys have heard the word before, a metabolism, someone having a fast metabolic rate or a slow metabolic rate. What is metabolism? So metabolism speaks to all chemical reactions that occur inside organisms. So any living thing that has chemical reactions occurring, all those chemical reactions collectively, they're called metabolic processes, or we call that metabolism. So whenever reactants are forming products, then that is a metabolic reaction. There could be simple reactions, there are complex reactions, but once they're occurring in living things, those are called metabolic reactions or metabolism. There are 2 metabolic pathways. So regardless of the type of chemical reaction, each type of chemical reaction can be further broken down into 2 metabolic pathways. Now, what are metabolic pathways? These are a series of chemical reactions that can either build complex complex molecules, or they're breaking down complex molecules. So any chemical reaction that is occurring inside of a living organism is either serving to make the products more complex than the reactants that are involved, or they're doing the reverse. They are making the products more simpler than the reactants. And we're going to get into that in a little bit. So here's an example of a metabolic reaction. So here we have a substrate, and a substrate is anything that can be catalyzed by an enzyme. So substrates, and then there is an intermediate product that is formed, and then there's another intermediate product that is formed, and then we have our final product. So we start off with our reactant, which is our substrate, and then that substrate changes into a molecule, and then it changes again into another molecule, and then we have our final molecule, which is our final product. And for each step, for each change that occurs, enzymes play a role, and enzymes are responsible for speeding up these chemical reactions. All right, so now we're going to talk about the types of metabolic reactions. So if you guys recall from the previous slide, a metabolism is pretty much any chemical reaction that occurs inside of a living cell or inside of living things. Um, Metabolic reactions, there are 2 types of metabolic reactions, or we can say there are 2 types of metabolic pathways. So the word pathways and reactions are used interchangeably. So the 2 types of metabolic reactions are going to be catabolic and anabolic. So regardless of the chemical reaction that's occurring in living things, it can be either or one of these. So we're going to start off with catabolic. A catabolic reaction is a reaction that releases energy. So energy will always be released in that process. And how is energy released? It starts off with reactants, because remember this is a chemical reaction. So chemical reactions have reactants, and then we're going to have final products. So the reactants are going to be complex. So these are going to be complex chemical molecules or compounds, and those complex chemical molecules or compounds will be broken down to form simpler products. So the end of the reaction, we're going to have simpler products than we started out with. And how do these products become more simple? Because bonds are going to be broken, and therefore energy will be released once those bonds are broken, and we're going to have simpler products. An example of a catabolic reaction would be cellular respiration. And I want, when you guys think of the word catabolic, I want you to think of the word catastrophic. And when you think of catastrophic or a catastrophic event, you're thinking like of a destructive event or something very devastating happening. Well, this is not as extreme as a catastrophic event, but when you think of catastrophic event, think of destruction. So these complex molecules will be broken down. In essence, they're almost like they're being destroyed, they're broken down to form simpler molecules. So that's just a way to remember catastrophic means breaking down. Anabolic is the opposite. So anabolic are types of metabolic reactions in which the opposite is happening, in which energy is not being released, but instead energy is being consumed. So a lot of energy is utilized in this reaction. So energy is actually needed in order for this reaction to even occur. And in this reaction, we're expecting simple molecules to start off. So our reactants are going to be simple. However, our final products are going to be more complex. So anabolic reaction is like building up molecules from simple to complex. And an example of an anabolic reaction would be photosynthesis. Photosynthesis takes very simple reactants such as carbon dioxide and water, and it builds those up to form complex products such as glucose. Glucose is a lot more chemically complex than carbon dioxide and water. So that is an anabolic reaction. In order to form glucose from carbon dioxide and water, energy is needed. So energy is locked in those bonds once those bonds are formed. All right, so one of the key things that AP Bio or College Board wants you guys to be able to understand. So this is like fundamental understanding. Even though catabolic reactions and anabolic reactions are different, they're opposing reactions. These two reactions are dependent on each other. So catabolic reactions can drive anabolic reactions, or anabolic reactions can drive catabolic reactions. So they're opposing reactions, however, they do depend on each other. So most anabolic reactions or most catabolic reactions, or let me say, most catabolic reactions actually rely on anabolic reactions. And I'm going to explain with this very simple diagram. So here we have a catabolic reaction occurring. Um, a catabolic reaction, here are our compounds. We have these compounds are broken down into simple ones. So as opposed to having one compound, we have one, two. Let me use my pointer here. So instead of having one compound, we have one, two, three. So we have three distinct molecules plus energy. These molecules were broken down. So the bonds were broken between these, and now we have three distinct molecules. Every time bonds are broken, energy is released. So that's how we have energy at the end. Now, this energy that is released at the end is now used to drive the anabolic reaction. So anabolic reaction will need this energy, and once this energy is provided, then anabolic reactions can now go ahead and build up complex molecules. So we have one molecule here, which is made up of three subunits. So the bonds exist between these. So we have one very complex molecule in our anabolic reaction. And when catabolic reaction occurs, this one molecule will be broken down into subunits or smaller molecules, and energy will be released. So the key takeaway from this slide is for you to understand that even though these are opposing reactions, they do depend on each other. So in this unit, there is going to be a lot of mention of energy. So let's just go through quickly what energy is. So energy is the ability to do work. Organisms need energy in order to survive and function. A loss of energy flow results in death. Whenever organisms completely lose their energy or cells do not have energy, cells die. And that's pretty much what death is just whenever our body no longer has energy. So if our brain cells are dying, we just don't have energy to fuel or to carry out the function of those cells, and therefore organs and cells will fail. There are different types of energy, and I'm pretty sure that you guys probably know a few of these or maybe all of these. But we're just going to go over this quickly. Types of energy and what those energies are associated with. We have kinetic energy, and kinetic energy is always going to be associated with motion. So whenever you think of kinetic energy, it should always be a body that is in motion or particles moving. So there's always movement involved, and that's what generates kinetic energy. Thermal energy is associated with movement as well, but specifically movement that generates heat. So thermal is associated with atoms moving, and whenever atoms move, heat is generated. So that's thermal energy. Potential energy is stored energy, so it's energy that is not currently being used for work. So think of like bonds that exist between molecules. Whenever there are bonds, whatever type of bonds they are, covalent bonds, whatever bonds exist between molecules, it means that the bonds, they hold the energy. So the energy is being stored. And whenever those bonds are broken, that is when the energy is released. And then we have chemical energy, and chemical energy is like potential energy that energy available for release in chemical reactions. So whenever the potential energy is actually released, we call that chemical energy. So whenever reactions are occurring and bonds are being broken, then we say that this is chemical energy because the energy is actually doing work. And these are the main types of energy that you guys need to know. All right, let's talk about the laws of thermodynamics. So we're going to go over the two laws that we need to know. And we're going to start off with the law of thermodynamics. So thermodynamics is the study of energy transformation in matter. So it's pretty much how energy is transformed from one form of energy to the next. And you guys should know that energy cannot be created or destroyed. However, energy is simply passed on from one form to the next, or is converted from one form to the next. And this law applies to the universe as a whole. All right, so what are the laws of thermodynamics? Let's look at the first law of thermodynamics. This law is the one that I just mentioned, which is energy cannot be created or destroyed. However, energy can be transferred or transformed from one form of energy to the next. So, for example, kinetic energy can become thermal energy. Chemical energy can become potential energy, or potential energy can become chemical energy. So, energy changes its form. However, energy cannot be destroyed, or can energy be created? The chemical energy or the potential energy that is stored within the nut that this squirrel is currently eating will be transformed into kinetic energy for the squirrel to climb the tree. So the energy that is gaining from consuming whatever food source is eating, well, in this case, you said it's nuts, that energy will be broken down. And once that energy is made available in the form of chemical energy, that energy can now be transferred as kinetic energy as the animal is moving. All right, let's look at our second law of thermodynamics. This law states that energy transformation increases entropy. And what is entropy? Entropy is disorderness of the universe. So as energy is changing form, some of that energy is going to be lost as other forms of energy. So energy is not uniformly changed from one form to the next, but however, we're going to have instability in energy, and also we're going to have energy lost. So during energy transfer, so as energy is changing from one form to the next, some of the energy is unusable energy because it is usually lost. And the primary way in which energy is lost is usually as heat energy. So as the squirrel is climbing the tree, some of the energy will be released as heat. So if the squirrel got 100% energy from the food that it consumed, it is not gonna, 100% of that energy is not available to climb the tree. 100% of that energy is not available for kinetic energy because some of that energy will be lost. So this is entropy. All right, so I've decided to add some misconception check slides in here. So it's pretty much for each slide that I'm going to be going through, and I don't want to spend too much time on it because I know that you guys can just pause the video and you can read. So there is a scenario, there is a question, and then there is an answer to the question. So it's pretty much highlighting some misconceptions that students typically have. So I'm going to pause the videos on each of the misconceptions, and I want you guys to read them, check the answer, and then if you guys have any questions, you can always ask those questions in class. Here's another one. Okay, so now we're going to talk a little bit about free energy. So this is also called Gibbs free energy, and it's named after the scientist that pretty much came up with this whole theory or this mathematical calculation for free energy. So his last name was Gibbs. Now, here is the cool thing. This is great news. Gibbs' free energy equation is now beyond the scope of AP Bio, and you're not required to know the mathematical equation for this, nor are you required to be able to do calculations for Gibbs' free energy. And this is great because when I was teaching AP Bio, we had to do this. So this was actually a part of Unit 3. But College Board decided to do away with this topic. However, the concept of free energy is still relevant to what we're doing. So the concept is what you need to know, not how to do the math part of it. So you'll never be asked how to do the math part of it. So I'm just going to go ahead and I'm going to explain what Gibbs free energy is. So Gibbs and whichever other scientists helped him, they came up with this math equation to determine the likelihood of a reaction occurring, a chemical reaction or a metabolic reaction occurring in a living thing, or if that metabolic reaction is energetically favorable. So is it within can it happen? Is there enough energy to drive this reaction? Is it feasible for this reaction to occur in terms of the availability of energy? So what Gibbs and whichever scientist he worked with that came up with this, they came up with this math equation in which you can determine the change of energy. So this is delta G. So this is the change in, and the G is for Gibbs. So this is a change in free energy is equal to the change in total energy minus, or you're subtracting the temperature. So this is heat energy, thermal energy, and the change in entropy. And if this is confusing, it is fine because we're not going to be studying this at length. But Gibbs free energy, or free energy, is pretty much surplus energy. So after the system or the body or the cell has utilized energy to do the vital, the important stuff, how much energy is actually left behind to do spontaneous reactions? So reactions that are occurring, not well, they're important reaction, but reactions that will just occur. Like for example, is there enough energy for cellular respiration? Is there enough energy to drive photosynthesis right now in the plant? Once all the other energy requirements are met, so all the energy requirements that are vital for the survival of the organism, once those energy requirements are met, how much energy is left behind to do spontaneous reactions? So what is the surplus energy that is left behind? You guys can skip this slide if you want to, if you don't care so much for a lengthy explanation of Gibbs free energy. All right, so a little bit more about free energy. So the free energy change of reaction determines whether or not a reaction can occur spontaneously. So the spontaneous reactions, no outside input of energy is required. So it means that if a reaction is occurring spontaneously, it means that no energy from an outside source, or no energy from generated from another reaction is needed. However, that reaction that is occurring will be providing its own energy. So whenever bonds are broken, so for example, whenever bonds are broken within the reactant, the energy that is released from those bonds will be used in that reaction. So that's what makes it spontaneous. So it requires very, very little energy for these reactions to occur. Now, based on the free energy changes, chemical reactions can be classified as exergonic reactions or endergonic reactions. And I know that most of you guys probably done this in chemistry, or you're about to do exergonic reactions and endergonic reactions. Um, Exergonic reactions are reactions that will release energy. So when you think of exer, so think of the prefix ex. So energy will be leaving, or energy will be released. And whenever energy is released, we can always tell when exergonic reactions are occurring. And I remember when I was doing chemistry in high school, we could just touch the beaker or the test tube, and we could feel the temperature on the test tube. So if we know if we're doing a chemical reaction and the test tube is now warm, then we know that that's an exergonic reaction because heat is given off. And it means that that energy is being released. So exergonic reactions are reactions that will release energy. For example, cellular respiration is an exergonic reaction. Um, Reaction is spontaneous. So exergonic reactions are spontaneous if the Gibbs free energy that is required is actually zero. So we know that that is a spontaneous reaction. So, and this is what it would look like. So this is Gibbs free energy. So this is the amount of energy that is being released from the reaction. And then this is the amount of energy that is actually needed by the products. So the products actually require a lot less energy. However, the reactants are giving off more energy in that reaction. So the Gibbs free energy is pretty much minimal. So it actually is a spontaneous reaction because it does not require, those products does not require a lot of energy. All right, so here is a reaction that is a endergonic reaction. Endergonic reactions are reactions that absorb energy. So, for example, for example, photosynthesis is an endergonic reaction. And again, when I was doing chemistry in high school, we felt the beaker or the test tube, and if it were an endergonic reaction, then the beaker or the test tube will be pretty much cold because a lot of energy was consumed from that reaction. And these are usually cool experiments to do. I don't know if you guys are doing that in chemistry. All right. So if the Gibbs free energy, if it requires a lot of energy in order for the reaction to occur, then it is not spontaneous. And photosynthesis is not a spontaneous reaction. And photosynthesis is an example of an endergonic reaction. And if you guys remember from previous slides, photosynthesis is also an example of an anabolic reaction. And anabolic reactions, they consume energy. And it requires more than free energy in order to drive that reaction. So there's a high demand for energy, and therefore that free energy is not enough to drive those chemical reactions, such as photosynthesis. So therefore, it is not a spontaneous reaction, and this is an endergonic reaction. All right, so here's something else that College Board wants you guys to know. So College Board wants you guys to know cells and their energy requirements. So why do cells need energy? Like, what is this energy being used for? So living cells have a constant flow of materials in and out of the membrane. The cells are not at equilibrium. So at any given time, cells are not always, or in most cases, they're not at equilibrium. Cells perform three types of work. So they're either doing mechanical work, so they're moving. Like, for example, the cilia is beating, and the cilia are the little hair-like projections that line cells in the lungs. So those guys are moving. Chromosomes also move during cell division. And then we have muscle contracting. So that's mechanical work. And also our cells are doing transport work. So they're pumping substances across the cell membrane. And as we know, active transport requires a lot of energy. So that's a higher energy demand. And then we have chemical work also occurring, in which we do have chemical reactions in which molecules are being synthesized. So molecules are, for example, we have building up of polymers from monomers, and that consumes energy. So these are some of the things that actually requires energy by the cell, because the cells do these constantly. Now, what supplies the energy is ATP. And ATP is an energy storage molecule. ATP stands for adenosine triphosphates, which means that there is an adenosine group and there are three phosphate groups attached to the molecule. And this is pretty much the universal energy storage molecule in all living things. And this is just a diagram, just a simple diagram just showing the structure of ATP. So we have three phosphate groups, we have adenine group, and then we have a ribose sugar group added to this ATP molecule. And this is pretty much how ATP is structured. Now, ATP, it couples exergonic reactions with endergonic reactions. So remember from a previous slide that we just discussed it, what exergonic reactions are and what endergonic reactions are. So the ATP couples these two reactions in order to do cellular work. Exergonic processes drives endergonic processes. And if you guys recall, exergonic processes, they release energy. So energy is always going to be given off in those reactions. Their initial energy input is very minimal, so it requires a very small amount of energy in the beginning. However, it yields a lot of energy. So, for example, think cellular respiration. The energy that is generated from cellular respiration is the energy that is used to drive endergonic reactions such as photosynthesis. So photosynthesis requires a lot of energy, and that some of that energy is supplied by cellular respiration, in addition to, of course, capturing the energy from the sun. So ATP pretty much utilizes these two processes. So organisms obtain energy by breaking the bonds between the two phosphates, phosphate 2 and 3 in reaction that called hydrolysis. And we know what hydrolysis is. We all know that hydrolysis requires water to break those bonds. So we're going to have hydrogen bonds being formed as opposed to whatever other bonds these molecules had going on. So whenever hydrolysis occurs, ATP is now broken down to ADP. And ATP, remember, ATP is adenosine triphosphate, and this is telling us that there are three phosphate groups attached. Whenever energy is needed, this ATP loses a phosphate and it becomes ADP, and the D stands for di. And di, the prefix di means two. So now we have a molecule that has two phosphate groups as opposed to three, because this third one is broken down, that bond is broken in order for energy to be released, and that's the energy that is used to drive these endergonic reactions. Um, Phosphorylation. So the release of phosphate moves to another molecule to give energy. So the release of phosphate moves to another molecule in order to give energy. When we talk about phosphorylation, we're talking about the process by which phosphates are being added to another molecule. That's pretty much it. So once phosphates are removed, energy is going to be released because that phosphate, the potential energy between those bonds is now no longer potential energy, is now being made available, and we have phosphate being added to another molecule, and that's phosphorylation. And we're going to talk about phosphorylation when we talk about our unit 4, when we're talking about cell communication. So hydrolysis of ATP. Hydrolysis of ATP is the process by which ATP is actually converted to ADP. And this molecule right here that's moving on the screen is, let me pause, you guys should figure it out. This is water. So hydrolysis involves water. So water is going to come here, and water will cause this phosphate, which is our second phosphate. So we have phosphate 1, 2, 3. This second phosphate, once water comes in the picture, this phosphate will be like, you know what, I want to bond with water as opposed to bonding with this other phosphate. And once water comes into play, um, hydrogen bonds will form, and then it will release this phosphate, and the energy will be released from that phosphate. So there we go. Phosphate is gone, and then we're going to have water joining at the end. And that is how hydrolysis occurs. Now, if hydrolysis occurs, so here's what happens. The rest of the molecule stays intact. However, we have this third phosphate that is now released, and that energy is also a product of that reaction because energy is also released. So energy comes from the lowering of the free energy, not from the bond itself. And that's something that's very important. So the bond that is broken, what the bonds will do is it will, what the molecule does is it pretty much lowers the amount of free energy that is required to drive the reaction. And we're going to talk about that when we start talking about activation energy. And that's going to make a little bit more sense in a bit. All right, so this is pretty much what I just discussed with you guys. I don't want to go over it again, just for the sake of time. So just look at this diagram, and once you look at it, and you read through everything, it will start making sense. All right, so all this time we've been discussing metabolic reactions in terms of energy requirements. So is energy required? Is it a spontaneous reaction? Is it a non-spontaneous reaction? And I just want to review a few things. If energy is required in order for the reaction to occur, it tells us a few things. So if energy is required, it tells us that, hey, this reaction is an anabolic reaction, because anabolic reactions, they consume energy. And also, if energy is inputted in this reaction, it means that this reaction is an endergonic reaction. And it also tells us that if this reaction needs energy, then it means that this reaction is non-spontaneous. So we've been talking about reactions in terms of their energy needs. Now we're going to talk about the rate of metabolic reactions. So how fast these metabolic reactions are occurring. Now I'm going to put some stuff on the slide here, and you guys can read through this slide, but I just want to explain something else so that I could just move on in the interest of time. So okay, so reactions either need energy or they don't need energy. A reaction that is supplied with energy does not necessarily mean that that reaction is going to occur fast. It does not affect the rate of the reaction so much. So energy can be supplied to a reaction. However, it does take a relatively slow time, or it still has a relatively slow rate. Now, what will increase the rate of chemical reactions or metabolic reactions are enzymes, and that's what we want to get to. So now we're going to start discussing enzymes. All right, so here we are, one of the big topics in Unit 3, enzymes. So we're going to talk about enzymes. We're going to try to describe the structure of enzymes and also what are some of the functions of enzymes. All right, so enzymes are macromolecules, and if you guys recall from Unit 1, enzymes are proteins. So they're made up of proteins. Proteins are types of enzymes, and you should know this. And this little diagram right here, let me get my pointer. This little diagram right here should have been a clue as to what enzymes are made up of. So they're made up of proteins. Enzymes are described as a biological catalyst, or they catalyze reactions. And when they by catalyzing, or when we say they catalyze or they're catalyst, it means that they speed up reactions. So speeding up a reaction is now describing the rate of the reaction. So how fast can these reactions occur? And enzymes allow for metabolic reactions to occur faster than they normally would in the absence of enzymes. So they speed up the reaction, and how do they do this? They speed up reactions by lowering the activation energy. So they actually lower the amount of energy that is required for this reaction to occur. All right, so let's highlight some facts about enzymes. Enzymes are not used up in the process. So enzymes are a key role in metabolic reactions. However, they are not reactants themselves, and they are not a part of the products themselves. So they're just agents there to speed up chemical reactions. So they're just catalysts. They're not going to be consumed in the process. Also, enzymes are a type of proteins, as I mentioned earlier, and enzymes usually end with A S E, so they're ACEs. Um, just a key thing, not all enzymes end with A. So, for example, most digestive enzymes, so trypsin, pepsin, like those enzymes do not end with ACEs, but any word that ends with an ACE is an enzyme. Not all enzymes end with ACEs, but any word, and all the ones that you're going to be exposed to in AP Bio, they will end with ACEs. All right, so now we're going to get into the structure of enzymes. So the enzymes, they act on reactants, and remember the reactant parts of a chemical or a metabolic reaction or a chemical reaction is what it starts with. So these are the things that are going to be reacting together in order to form products. So they act on reactants and not products. Reactants, whenever we're describing reactants in this context, in the context of enzymes, we call the reactants substrates. So substrates are any reactants that the enzyme specifically acts on. All right, so for the sake of a diagram, this is an enzyme. Now, enzymes are proteins, so we know that they're going to be coiled and convoluted. They're going to be wrapped in or folded in different ways according to whatever side chains or R chains they have, and they could form up to the tertiary or the quaternary structures. But for the sake of a simple diagram, we're going to say that hey, this is a model of an enzyme. Now, an enzyme has what is called an active site. And here's our active site. So it's in this region in here. The active site is the area where the substrate will bind. So in other words, the reactants are going to be binding to the enzyme right here. Now I want you guys to think, because this is AP Bio and we did Unit 1, we know that enzymes do not look like this purple blob. So whenever we talk about active sites, I want you guys to think of proteins. So it's going to be a part of the protein that is involved in the chemical reaction. Now, what part of proteins are involved in the chemical reaction? The R chains. So each enzyme will have a specific R chain or R chains in which their reactants will bind to or form reactions with. All right, so in describing the function of enzymes, so we have established what the active site is. We know that enzymes are proteins. So how does this work? Now, substrates will bind to the enzyme at the active site, and once the substrate and the enzymes are bounded, we now call this an enzyme substrate complex, and that's where the reaction will occur. The enzyme will cause the substrate to now form products. So the substrates in the reaction will now form the products of the reaction. And then once those products are formed, those products, they leave the enzyme. So they leave the active site of the enzyme, and then the enzyme itself is now available to do pretty much the same thing in other similar reactions. So a key thing about enzymes are not depleted, enzymes are not used up in the product, in the process. Enzymes can be denatured. However, enzymes are not consumed by the reaction that they're taking part in. So let's go through this one last time. So here we have an enzyme with the active site available. Here we have the substrate. These substrates are perfect fit for the active sites. Once substrate binds to the enzyme at the active site, we call this complex with the enzyme and its substrate an enzyme substrate complex. Enzymes will cause these reactants to now become new products. Once these new products are formed, the products will now separate from the enzyme, and the enzyme is now available pretty much to break down other similar substrates. And it will go on and on and on as long as substrates are available. And enzymes are in a conducive environment. They will just be reused over and over again. They're not going to be used up in the process. There are different models of how enzyme substrate complex are formed. This is the most basic model. So this is what we call the induced fit model. And there's another model that is called the lock and key model. I don't have it on this slide, but let me just explain lock and key model really quickly. So lock and key model is pretty much when an enzyme and the substrate, the enzyme's active site fits the substrate perfectly. It's a perfect fit. So it's a lock and key fit. That is what lock and key is. Now, there's another model in which enzyme substrate complexes are formed, and that is called the induced fit. So on this slide, I'm describing now the induced fit. The induced fit model is when the substrate, which is this orange shape right here, the substrate is not a perfect fit for the enzyme's active site. However, the enzyme can pretty much change the shape of its active site to better fit the substrate. So it's being induced in order to fit the substrate. And remember, these are proteins. So by an induced fit, a protein can pretty much change the way it is folded, not any great change, but just very small modifications in order to be able to bond with the substrate. And that is called induced fit. So once we have an induced fit, we're going to have the products now formed. So if you guys follow this, we're going to have products formed at the end of the reaction. And sometimes enzymes are performing catabolic reactions. So this is enzyme catabolism. If it is catabolic, and I want you guys to pause, think about it, what type of reaction would this be? Hmm. All right, so when we talk about catabolism or catabolic reactions, remember catabolic reactions, catastrophic, something is being broken down. So here we have a complex molecule going into the active site of the enzyme, and at the very end of this reaction, we don't have just one molecule, but instead we have two. So this one molecule that we started out with was broken down to now form two molecules. So again, broken down. That is a catabolic reaction, or that is catabolism. And also, you guys should know, if your minds are working, you should know that, hey, this is going to be releasing energy because it is catabolic. So once bonds are broken, energy is released. All right, how do enzymes perform anabolic reactions? So enzyme anabolism. So again, pause, think about it. Anabolytic reaction, or anabolism, is pretty much the process by which we're going to start off with simple reactants, and then at the very end we're going to have more complex reactions. And that's anabolic reactions. All right, now we're going to discuss the effects of enzymes. So, enzymes are proteins, we keep saying that, which means that it has a 3D shape, and we should know the 3D shape of proteins. So, we're expecting it to be the tertiary shape, and it can be affected by different factors. So, what are some of the environmental factors that could affect proteins, or just enzymes? The efficiency of the enzyme can be affected by temperature, pH, or chemicals. So other chemicals can also affect the temperature. And I want you guys to always think of this. Enzymes are proteins, and the shape of a protein determines the function of the protein. So the protein has to be folded in a specific way, and enzymes are very specific to their substrates. So if the temperature affects the folding of the protein, so if the protein starts to unravel, then it means that that enzyme is no longer capable of doing its job. Also, the pH, so whatever pH the environment is, can also affect how these R chains interact, and it affects the folding of the enzyme. And also other chemicals, maybe some chemicals can compete for the active site. So sometimes enzymes will favor other chemicals over their actual substrates, and this also affects how well the enzymes work. Or sometimes chemicals can destroy the enzymes themselves depending on what those chemicals are. And here's something I always tell you guys to remember. Proteins are very specific. So the shape of the protein determines how it functions. Think of enzymes, guys. Enzymes are specific to their substrate. The active site has to be a perfect fit, or it has to be slightly modified in the induced fit in order to fit its substrate. The change in the shape of the enzyme will change how it functions. So if enzyme shape is changing, it might cause the enzyme not to work at all. All right, so again, we're still talking about effects of enzymes, or effects of environmental conditions on enzymes. So here's a very key thing that we have to know. What are optimal conditions? Optimal conditions are the best conditions at which enzymes actually work and function. So these are what are the ideal conditions for specific enzymes, and enzymes differ. Not every enzyme requires the same conditions. Some enzymes function best in acidic environments. So for example, pepsin in the stomach. The stomach is a very acidic environment with hydrochloric acid in your stomach, and that's how pepsin best functions. Some enzymes, they require more alkaline or basic environments in order to function. So, for example, trypsin in the small intestines require a more alkaline environment or a more basic environment in order to function. Now, those are what we call optimal conditions. So, what is the favorable temperature for each enzyme and the favorable pH that allows enzymes to work best? and it differs from enzyme to enzyme. The rate of the enzyme activity increases once it has reaches optimal, anything, optimal temperature or optimal pH. So the rate of enzyme activity increases with temperature due to collisions up to a certain point. And this part is important, so I'm gonna highlight this. High temperatures are good, or increasing temperatures are good, and up to a certain point, because temperatures can get high to the point where the enzyme becomes denatured. And when we talk about denaturing of enzymes, we mean that the enzyme is now not folded correctly, or it affects the folding of the enzyme, and therefore that protein does not work anymore because it has now unraveled, or it's not folded correctly. Also, enzymes will function at a specific pH, and at that specific pH, the activity of the enzyme will increase. However, it starts decreasing once that pH is no longer favorable. So if enzymes are outside of their normal pH, you're either going to be working at a very slow pace or slow rate, or just not at all. So they're probably just not going to work at all. So just remember this word denature. It means that the enzyme is destroyed, and being that enzymes are protein, it just means that the shape of the protein is now not what it should be, and therefore it does not function. All right, so this slide is pretty much showing you three different enzymes. So these are just like generic enzymes, enzymes 1, 2, 3. So they're not specific enzymes. On your AP Bio exams, they will give you specific examples of enzymes. You do not have to recall any enzymes by name or what their optimal temperature or pH is. That is beyond the scope of AP Bio. You have to have basic fundamental understanding that, hey, whenever they're at their optimal, they will function best. So if this is the rate of the enzyme reaction and these are different temperatures, then we know that enzyme 1 functions best at 20 degrees, and we know that enzyme 2 functions best at around 50, 55 degrees, and we know that enzyme 3 functions at around 85 degrees. So optimal is the highest point on the graph. Everything before it reaches the optimal is going to be performing very slowly or not at all. And everything after it reaches the optimal is going to be very slow or not performing at all. All right, so now we're going to talk about enzymes and cofactors. So these are very important things. So enzymes, oftentimes they require other molecules to pretty much enhance their performance. So it's almost like they're on performance enhancement. So enzymes do a very good job at speeding up chemical reactions, but sometimes the presence of other things, other molecules can also help that enzyme to better function. So we call these things cofactors. So cofactors are molecules that assist enzymes in functioning. And what I want you guys to know, and I'm going to use a different color for this, for those of you guys who are listening to this video, cofactors are not proteins. So these are not proteins themselves. So any non-protein molecule that assists an enzyme or causes enzymes to work better, or causes enzymes to better speed up reactions, those collectively are called cofactors. Cofactors are going to be inorganic, and we should know that if they're inorganic, then they're not carbon compound. And examples of these could be metals. Um, can be bounded very loosely to the enzymes, or it can be bounded tightly to the enzyme itself. It's not going to be at the active site of the enzyme, so that's something that's key. So it's going to be bounded to the enzyme somewhere else and not on the active site. We have holo enzymes, and these are enzymes with cofactors attached. So these are enzymes that already have their cofactors attached. So that's just how they are. They have their cofactors already, almost like it's a permanent part of the structure of the enzyme. And then we have some of them that will, they can be enhanced whenever the presence of these inorganic factors are there, or so it's like if the factors are there or not. And then some of them, they do have that already. It's almost like already built in. All right, so those are cofactors. So now let's talk about coenzymes. Coenzymes are organic. And this means that these guys are going to be like carbon compounds. Examples of these are vitamins, and pretty much, yeah, that's the example. That went by too fast. All right, enzyme inhibitors. Now, there are some other molecules or other chemicals that can pretty much prevent enzymes from working. So they inhibit. So this word inhibit means to prevent. So they're inhibiting enzymes from doing their job. And how do they do this? They pretty much could compete with the enzymes for the active site. So enzyme inhibitors, what they primarily do is they reduce the activity of a specific enzyme. So inhibition can be permanent or it can be reversible. So sometimes they will stop the enzymes permanently from working, or they will temporarily stop the enzymes from working, and the enzyme can resume working once the inhibitors are removed. Permanent inhibitors bind with the enzymes, forming covalent bonds, and covalent bonds are very strong bonds, hard to break. So those make the enzymes permanently prevented. It's prevented permanently from doing its job. So those are permanent inhibitors. And examples of these are toxins and poisons. So some toxins and poisons, they prevent enzymes from doing their job, and therefore they can be very lethal. Because if enzymes don't speed up reactions, depending on what those reactions are, they could literally cause death. Reversible enzymes, they bind by other weaker interactions, so not covalent bonds, and those interactions can be easily broken. All right, so enzyme inhibitors. Some inhibitors are competitive inhibitors, and some inhibitors are non-competitive. So we're going to go over what these two things mean. So think of what I just said from the previous slides. What are inhibitors? They can be permanent, they can be reversible, and now we're going to talk about types of inhibitors. So we spoke about what they are, and now we're going to talk about what types they exist in. So inhibitors can be described as competitive inhibitors. And all inhibitors, regardless of the type that they are, they will reduce the activity of an enzyme or prevent an enzyme from doing its job. Now, we describe inhibitors as being competitive if inhibitor is actually blocking the active site of the enzyme. And if you recall, the active site is where the substrate binds. So if the inhibitor comes here and blocks this active site by binding with the enzyme at the active site, then it means that the actual substrate is unable to bind at that active site because the active site is already occupied with the inhibitor, and therefore the enzyme just does not work. Inhibitors can be reversible, so they can be reversed with increased substrate concentration. So if there's a lot of substrate in that environment, then the inhibitors pretty much can be out-competed, if that makes sense. So if there's more of the substrate than there are inhibitors, then it means that the inhibitors are out-competed because there's more of the substrate to bind to these enzymes. Here we have just examples. We have, this is what the substrate should look like, and this is the active site. Substrate binds the enzyme. That's normal. Everything is good. However, if an inhibitor, a competitive inhibitor, goes and blocks that active site, then it means that the substrate is not able to bond to the enzyme, and therefore the enzyme is not able to break down that substrate or build it up, depending on what the substrate was. Here's another type of inhibitor. We have non-competitive inhibitors. And these guys, they still inhibit, so they still prevent the enzyme from doing their job, but they're regarded as non-competitive. And here's why these guys are regarded as non-competitive. Because they attach to the enzyme at another site, so not at the active site. So they will bind to the enzyme somewhere else, but not at the active site. So they're not blocking the active site. And these types of inhibitors are called non-competitive because, again, they are not competing for the active site of the enzyme. Now, they bind to the enzyme, however, they bind somewhere else. And the site in which they bind is called the allosteric site. So they're pretty much binding to the enzyme somewhere else other than at the active site. And remember, enzymes are proteins. So if something else is there's another chemical that is binding to the protein, that will now change the shape of the protein. And if it changes the shape of the protein, it changes the function of the protein. So once we have something binding to our protein somewhere else, that changes the shape of the protein, and by therefore doing it, it's changing the shape of the active site of the protein, and it's preventing the substrate from binding. So, in simple terms, even though non-competitors do not compete for the active site, they're binding to the enzyme somewhere else, and they're causing the enzyme to become distorted. So the shape of the active site no longer fits that substrate, and therefore the substrate, the enzyme substrate complex is not formed because the active site is available, however, it is not the right shape anymore because of this non-competitive inhibitor. And here we have an example. So the active site is here. However, our non-competitive inhibitor is binding somewhere else. And remember that somewhere else on the enzyme is called the allosteric site. And it changes the shape of the protein, and therefore protein does not work. All right, so chemical reactions can be regulated. So a cell must be able to regulate its metabolic pathways. And how do cells do this? Cells can control where and when enzymes are active. So they can actually control when the enzyme is active, where the enzyme is active. There are specific genes that can switch on or off an enzyme. So there are genes that can prevent enzymes from working, or they can temporarily prevent an enzyme from working. All right, so we're going to talk about allosteric regulation. So we should know that allosteric means that there's another site in which the enzyme, a molecule is going to be bonding to the enzyme. And we're talking about regulating enzymes. So how enzymes should not just be working all the time, just constantly speeding up reactions. There are some times when our body needs these metabolic reactions to stop. So all metabolic reactions are not just ongoing reactions that our bodies need all the time. There's sometimes when these reactions just occur as needed, and if it's not needed, then it should be switched off. So allosteric enzymes must have two binding sites. So allosteric enzymes, so these are enzymes that can be switched off. So whenever we describe an enzyme as an allosteric enzyme, it means that it has a place where another molecule can just pretty much go and bind and switch it off. So allosteric enzymes must have two of these binding sites. It must have the active site, and the active site is for the substrate. In addition to the active site, it also has an allosteric site, and this is the site where regulation will occur. So this site is any site on the enzyme other than the active site. So, and it's very similar to what we described with the non-competitive inhibitor. So here we have an enzyme and its active site, and here we have the allosteric site. And this is another example. Here we have active site of enzyme and we have allosteric site. This is an enzyme, and this is a multi-unit enzyme. So this is also an enzyme. This is showing an enzyme that is able to break down more than one substrate at a time. So the allosteric site is always going to be somewhere else that is not the active site. Allosteric regulation, what happens is these are molecules, and these are molecules that will form non-covalent interactions, which means that these are temporary. These are not strong bonds. So molecule binds to the allosteric site, which changes the shape of the protein, and then by doing so, it changes the function of the protein. So once it changes the shape of the active site, it means that the protein no longer functions the way it should because the active site has a different shape. This may result in inhibition of the enzyme, so it pretty much acts as an inhibitor. Or sometimes they could be stimulators in which they're actually activating the enzyme. So it depends on what type of enzymes it is. So allosteric regulation can actually be the dual purposes. So it can serve to switch the enzyme on, or it could serve to switch the enzyme off. And pretty much how it works is if it's allosteric regulation can be activators or deactivators, or inhibitors, sorry, activators or inhibitors. So if it is activating, it means that only when the allosteric molecule is attached to the enzyme will work. So in order for the enzyme to actually function, the allosteric molecule has to be attached. And that's pretty much what it means. I'm going to leave you to read over this slide, pause it, read over it. I know that once you guys read this, you can understand it. And then I'm going to go to the opposite, which is allosteric regulation that results in inhibition. All right, so here we are. So the opposite would be of another type of allosteric regulation is when the molecule or the inhibiting molecule actually serves to stop the enzyme from working. So once that molecule is placed at the inhibition site or the allosteric site of the enzyme, it means that the enzyme is now rendered deactive. So the enzyme does not work, it's now inactive. Um, and that's pretty much. All right, so let's talk about a unique type of allosteric regulation. So this is cooperative. And cooperativity is pretty much when the substrate itself serves as the molecule that regulates the enzyme. And this is unique because it happens only in enzymes that have these multi units. So this is an enzyme. So these are examples of enzymes. Cooperativity is when the substrate binds to one of the active sites of the enzyme, and by doing so, it pretty much opens all the other active sites. And if the substrate is removed, then pretty much all the active sites are closed. So in this example, it is unique because that's not how all enzymes are, but this is when the substrate itself is functioning as the molecule that switches the enzyme on or off. And we call this cooperative or cooperativity. And this is considered allosteric regulation since binding at one side changes the shape of the other side. So that's why this is categorized as allosteric. All right, so for this last slide, we're going to be discussing feedback inhibition. So this is also a type of inhibition. So this is a type of inhibition in which the products of a metabolic pathway serves as an inhibitor for another metabolic pathway. So the products, what is produced at the end of a chemical reaction, serves as a inhibitor for other enzymes in a similar or same pathway. So this is a way of, and this is called feedback inhibition because this is a way of like stopping up the body or the system or cells from overproducing something. So it's almost like saying, hey, we have enough of this, so do not continue to do this. So we're not trying to overproduce this specific process. All right, so I'm going to use my little diagrams here to see if we can make sense of this. So here we have enzyme 1. So this is our enzyme. The red part is our enzyme, and then the green is our substrate. So enzyme 1 produces an intermediate molecule is produced, and then enzyme 2 is responsible for changing that intermediate molecule into a product. So this is the end of our reaction, right? So here we went from our triangle substrate, and now we have another enzyme that does play a role. So this required two enzymes, and then we have the desired product at the end, which is a circle, a yellow circle. Now, if this is a feedback, it means that this said enzyme, so see this enzyme is the same shape, this enzyme will no longer continue to break down triangles and form circles because the circle, which is the product of this reaction, is now being used as an inhibitor. So this enzyme has an allosteric site. So this is pretty much, there are a few chemical reactions. I wouldn't say a few, let me not quantify them, but there are chemical reactions in the body that does not require overproduction of a specific product. So a mechanism to avoid overproducing something is to use the product itself as an indicator that, hey, we have enough of this, do not continue making it. And pretty much once that happens, then pretty much the pathway pretty much stops. It will continue again. So this is not permanent. So it will continue once this product is pretty much absent. So once that product has been utilized, has been used up, or it has been transported somewhere else, this would be the indication to open up back this metabolic pathway again once that product is no longer present. All right, this is a free response question. I don't want to spend too much time on this. Maybe we're going to do this in class, maybe not. But this is a free response question. You guys can pause and try to answer the questions. Here's another practice question. You guys can pause and answer the question. All right, we've pretty much come to the end of this lecture. I know it's long, guys. I know it's a long lecture, but here's the thing. We have covered most of Unit 3 in this lecture. So we have covered topics 1 to 5. We only have two more topics to do, and that's photosynthesis and cellular respiration. And I'm going to divide those each into two videos. So I'm going to do a video for photosynthesis, and I will do a video for cellular respiration. But we have covered, maybe I would say this is about 60% of Unit 3 that we have covered so far.